Welcome, everyone, and thank you for joining us wherever you are in the world. Good morning, good afternoon, and good evening to all. My name is Brad Pesaveno, Senior Product Specialist in Thermo Fisher's Scientific Materials and Structural Analysis Division with electron microscopy. I'll be your moderator for today's session. Ask the Experts is our monthly series that focuses on various electron microscopy topics with an interactive discussion on microscopy applications and techniques. The series started in 2020, and to date, over 20 sessions are available to view on demand. Please be sure to sign up to receive our emails or visit our Ask the Experts website on our Learning Center to receive notices about future sessions. A few housekeeping items before we begin. This session is intended to be an open forum to provide answers to your questions. These sessions will be recorded for later on-demand viewing. To ask a question, please use the Q&A box and send to all panelists at any time during the webinar. This is the only way to ask a question since all lines will remain on mute. We'll take as many questions as possible live, but if we can't get to your question in the allotted time, we will reach out via email to contact you directly. We're also always looking to improve, so any feedback on future topics is welcome. Today's experts. I'm happy to introduce our experts for our session on Cryo-EM meets AI, bringing innovation to drug discovery. First up, we have Dr. Xiaodan Ni, Senior Scientist in Cryo-EM and Structural Biology with Shuimu Biosciences. Xiaodan obtained her PhD degree in structural biology at McMaster University in Canada. Afterwards, she went for postdoctoral training at MRC LMB in the United Kingdom and at the NIH in the US, working on mitochondrial outer membrane translocons and Parkinson's disease-related alpha-synuclein fibropolymorphism. She joined Shuimu Biosciences in January 2021. Next up is Dr. Pan Li, Artificial Intelligence and Structural Biology with Shuimu Biosciences. Pan got his PhD degree in biology from Tsinghua University in 2021. His research lies in the study on RNA secondary structure, AI, and protein structure. He joined Shuimu Biosciences in July 2021 and is focused on protein structure and small molecular drug design. Rounding out our panel of experts, we also have Raymond Shriver, Senior Director of Strategic Technology, and Edward Pryor, Director of Product Management at Thermo Fisher Scientific. We've got some great Cryo-EM experts today, so don't miss your chance to get your questions answered. I'll now pass it over to Xiaodan and Pan to briefly discuss some interesting workflow preparation and how they're using AI as part of our model building and future directions. Xiaodan, over to you.
Yeah, okay. Hello, everyone. Yeah, can you see my slides? Yeah, hello, everyone. I'm Xiaodan. It's a pleasure for me to have the chance to share our work in this webinar. So first of all, I will have a brief introduction of Shuimu Biosciences. Shuimu Biosciences, founded in 2017 at Tsinghua University, we are a core team of outstanding life and computational scientists, AI and pharmaceutical industrial experts. And we have actually successfully provided structural biology and drug discovery service for more than 100 innovative pharmaceutical companies and top research institutes from all over the world. And our purpose is to drive change to empower drug discovery and accelerate the development of innovation drugs. To achieve this goal, we leverage the power of cutting-edge AI and Cryo-EM technologies to help our partners better understanding structure of the target proteins and complexes. And as most of us know, when the first high-resolution structure of triple ion channel was solved to 3.4 angstrom in 2013, it really revolutionized the structural biology field because it proved that by using Cryo-EM technology, we have capability to solve the membrane proteins that are always resistant to X-ray crystallography. Then we went into the decade of resolution revolution. Of course, these achievements is due to the development of both hardware and software of Cryo-EM technologies, such as the direct electron detector and image processing softwares. Then the resolution levels in decades went from 2.2 angstrom beta galactosidase to nowadays we can solve the mouse evaporating to 1.2 angstrom. So by using Cryo-EM technology, we really achieve the atomic resolution. Then proteins smaller than 100 kD can be get to high resolution as well. From the 93 kD, acid citrate dehydrogenase was solved to 3.8 angstrom to nowadays the smallest 52 kD stratigraphy was solved to 2.6 angstrom. So the resolution development really provides us this important structural information to study all kinds of biological questions. And for example, the protein and peptide complex is useful for the following structure-based drug design. And the structures of antigen-antibody complex is useful for the conformational epitope mapping. And the structures of virus and virus-like particles can be used for vaccine development. In Suimu Bio, we actually have all kinds of experience for all these projects. And this is one of the cases we use Cryo-EM technology to study the structure of antigen antibody complex for conformational epitope mapping. So CD73 is the cell surface nucleosidase that generates adenosine and with the help of CD39. And in the tumor microenvironment, the accumulation of adenosine will always cause immune suppression through the regulation of cytotoxic T cells and NK cells. Therefore, by antagonizing the CD73 function offers a pathway to induce anti-tumor immune responses. So there's always two conformations of CD73, the inactive open state and active closed state. So currently, there's two antibodies against CD73 that are under clinical phase 2 and preclinical study has been identified to have severe WHO effect as the increase of concentration of antibody. So the epitope mapping experiment has identified that only when a specific binding mode appears that will allow to stabilize the inactive open state of the CD73. So in this case, when the concentration of the antibody increased, it will finally dilute the specific binding mode and cause WHO effect. So antibody that can avoid WHO effect is highly demanded. And in Suimu Bio, we actually collaborate with our client and find new mechanisms of antibody that bind CD73 through a unique intradimer mechanisms. Different from other antibodies, this new kind of antibody binds C-terminal of the CD73, and actually we can see the effects from two antibodies combine simultaneously to the CD73, thus breaking the concentration limit and avoid WHO effect. So this is just one of the applications of Cryo-EM technologies. And in our Cryo-EM platform, we also can develop the graphene oxide and reduce graphene oxide Cryo-EM increase, which called Graph Future. So I think as many Cryo-EM scientists should know, the sample preparation is the critical step for make sure that the quality of your data set. So you may encounter many issues such as your protein are damaged by the air-water interface and you have preferential orientation, or even one step ahead, your protein expression level is very low, so you can't get enough protein for the Cryo-EM study. So by applying a very thin layer of graphene membrane on the EM grids, you will increase the number of particles per image. And also, you will protect your protein from damage from the in the air-water interface, and also increase the random orientations. So this is basically the device we use to produce the GO and RGO membrane. So briefly, the graphene oxide solution will gently pipette on top of the water bath surface. Then you will disperse to form a graphene oxide membrane in the air-water interface. And the EM grids has been placed in the mesh sphere standard and immersed in the water bath in advance. So as the water slowly drained and the graphene oxide film can be coated in the EM grids and air dried. So the graphene oxide grids can be finally put in an atmosphere of hydrogen and argon plasma flow at 300 degrees for 1 hour to produce the reduced graphene oxide. And this is typically how the holes and the electron diffraction pattern of the specific area of our GO and RGO grids looks like under TEM. So compared to the graphene oxide grids, we also have the reduced graphene oxide grids. The reduced graphene oxide membrane actually has decreased the interlayer space with lower background noise. That means that you can increase the contrast of your particles. And you also enhance the electrical conductivity. That means you can decrease the charging effect and beam-induced motion to improve your image stability. And for our experience, we also find that the RGO membrane has a very nice particle absorption activity. That means you can avoid the damaging of your particles from air-water interface. And more importantly, the RGO supporting film is particularly useful in cryo-EM reconstructions of sub-100 kD proteins, such as the ACE2 and RBD complex can be solved to 2.8 angstrom by using the RGO membrane. So as a summary, the RGO membrane can be used as a robust graphene-based supporting film in cryo-EM sample preparation. Okay, so now I will pass the presentation to my colleague Pan for the AI application in Mobile. Thank you. Pan, go ahead.
Okay. The process of single particle analysis can be described in 3 steps: sample preparation, data acquisition, and image processing. Single particle analysis, thousands of macrographs with particle projections are collected from the Cryo-EM equipment. Particle picking is the first step in the computational pipeline of image processing. To improve the signal-to-noise ratio, particles are picked and averaged, and then used to reconstruct the 3D density map. The 3D density map reveals the detailed information of the specific macromolecule. The next step is to use the 3D density map to build the atomic model. Most of the steps in this workflow are highly automatic with known tools and methods. But model building often involves substantial human interventions, which is labor-intensive and time-consuming, especially for de novo structures. As we know, DeepMind released AlphaFold last year with impressive results and great potential for protein structure prediction. We leveraged the power of AlphaFold 2 to develop a new method called Cryo-Fold to automatically build the atomic model from the 3D density map. This method has 3 features. Firstly, it is automatic. The user inputs the 3D density map and the sequences, and the program uses the sequence to search the MSA and the PDB templates and combine it with the density map to build the atomic model. Less human intervention required. Secondly, it improves efficiency. Reduce the time of model building from a couple of weeks to several days. Thirdly, ideally, it can build a highly similar model, even if a much lower resolution map was used. That is to say, less raw data required. So in this way, you can reduce your cost for data collection and image processing. Except for model building, we will be developing other tools to facilitate the Cryo-EM data processing. For 3D reconstruction, we are developing CryoNet VAE to directly reconstruct the continuous distribution of 3D density map from the 2D particle images. CryoNet ET is used to refine the density map and improve its resolution. CryoNet resolution is a tool to estimate the local resolution of the density map. Cryo-Fold has been introduced in last slide. After the atomic model produced, CryoNet Refine is used to refine and fix the placement of the atoms, such as the side chain orientations, to improve the structure fitting. Based on our CryoNet platform, we believe AI technology would play more and more significant role as the extraordinary growth of Cryo-EM data produced in free mobile sciences and other facilities in the future. Questions are welcome.
Thank you, Xiaodong and Pan. This is a great segue from Cryo-EM to discuss where AI can go for all aspects of Cryo-EM. So now we're ready to move on to the Q&A segment of our webinar, and we already do have some questions that have come in. But just as a reminder, in order to ask a question, please use the Q&A box in your WebEx portal and click send to all panelists. That way we'll get all the questions and we'll try to answer as many as we have time for. But let's go ahead and get started with the first question. First one up, I'll direct to you, Xiaodong. How long does it take normally to build a model using Shuimu's solution?
Well, I think it depends. We have the simple case and difficult case. If the simple case, for example, people have already solved the structure and you can find the PDB, so it was much easier for you. You just fill the PDB into a density map and do a model building. But especially for some difficult case, for example, if you are de novo structure determination, so you need to use the AlphaFold. That is the usual way we do. We use AlphaFold to have an initial model, and based on initial model to build a final model. But now we have the AI-based QuantNet, just as Pan mentioned, right? So we can limit the time from 2 weeks of model building to maybe 2 days. Yeah, so that is benefit from our QuantNet.
Oh, great. Thank you, Xiaodong. Next question that's come in, this is one maybe for a couple of our panelists, but we'll start with you, Pan. Will AlphaFold 2 replace Cryo-EM or X-ray crystallography in the future?
Okay, this is a good question. AlphaFold 2 and AlphaFold database provide good resource for this field. But in my opinion, it is unlikely for AlphaFold 2 to completely replace the experimental methods such as Cryo-EM and X-ray crystallography. I have several reasons. First of all, the AlphaFold 2 paper reported that the distribution of significant score of those proteins with no homolog in the database is much lower. And AlphaFold 2 also requires the query-seek has as many homologous sequence as possible. That is to say, the AlphaFold 2 is not suitable for those proteins have no homologous sequences, such as human-designed protein or orphan proteins. Secondly, AlphaFold 2 often output structures with notable disordered regions, but it unknown these disordered regions are truly disordered regions or just failed predicted regions. Many disordered regions become ordered upon binding, and that is an important aspect of their functions. Thirdly, AlphaFold 2 is trained on the monomer dataset. Although DeepMind released a new version of AlphaFold, which trained on the multimers, but from our experience, the performance of AlphaFold multimer is much lower. More importantly, AlphaFold multimer predicts the complex structure based on the evolution information, which restricts it from predicting the antibody-antigen interactions. Finally, the AlphaFold 2 only output single conformation for each query protein, which limits its usage scenario. But a single particle analysis is able to resolve the conformational and compositional heterogeneity. Summary, I think AlphaFold 2 is more like an advanced homologous structure modeling tool. Its performance highly depends on the evolution information from the MSA or PDB templates. So I think it is unlikely for AlphaFold 2 to replace the experimental methods.
Thank you, Pan. That was a great, detailed response. I appreciate it. Raymond, maybe I'll bounce it over to you to follow up on that in terms of the benefit that the experimental techniques provide to some of the predictive methods. I know you're involved in some of our software development.
Absolutely, thanks. And I think the answer was already very complete. But indeed, what I see is that it will not replace, as was already mentioned, right? What it does do, AlphaFold will help make structure determination experimentally quicker and easier. And I think it was already shown in a great way how the model building and the 3D EM density calculation is greatly helped. We can also see that the construct design, the biochemistry side of things, is greatly improved if you upfront know what kind of structure you're going to get. And internally, we're using also AlphaFold and other AI methods to optimize the data acquisition itself. So indeed, all three of the parts, construct design, the sample preparation, the data acquisition, as well as data processing, will be improved by using AlphaFold. That in addition to the answer of already given.
Okay, great. Thank you, Raymond. Speaking about sample prep, this question came in, and Eddie, you might be able to help with this one. This is regarding small molecules. If a protein is small, say less than 50 kilodaltons, what type of work can we do with Cryo-EM? What are some of the things we can do with those smaller molecules?
Brad, that's a really great question. The common experimental techniques that are usually tried are just seeing how we can make a protein bigger, and that's either binding of antibodies or binding of Fabs or adding fusion proteins to the molecule of interest. And I think, if in the context of this discussion, talking about AI and its application to the Cryo-EM workflow, I think, there's a really nice example, and we talked about complementary techniques of AlphaFold or 3D structure prediction and Cryo-EM. And I think this is a particularly nice example where you can, a priori, before you even put things or even start preparing samples for Cryo-EM, start to use AI structure prediction techniques like AlphaFold to predict structures of either protein complexes and seeing how things might bind and make your sample bigger, or fusion proteins, for example. Where a fusion protein, let's say a C-terminal or N-terminal fusion protein, how that will interact with the sample of interest. So you could start getting some information about the sample before you even put it into the microscope. And I think AlphaFold is a great tool for starting to think about how we just take small proteins and make them bigger by adding protein complements to them.
Awesome, thank you. Thanks, Eddie. Xiaodong, you had mentioned in your part of the presentation you were using a graphene substrate, and we had a question come in from Monica Dunkel. Are the graphene oxide grids commercially available? Where are you getting them from?
Yeah, so actually based on our the flow of our production. So actually we put the graphene grids, the quantifoil grids with the graphene based support. So we can help our clients to coat their EM grids with this membrane. Yeah, so graphene and graphene oxide. So it's ready now. So we are happy to have our client to ask us to coat the EM grids with the membrane. Yeah.
Oh, great. Thank you. Thanks. And then Pan, you had talked some about CryoNet, and a question that came in from Legal Wang is, does the CryoNet performance depend on the map resolution? And do you have any statistics for that?
Sorry, I don't hear. Please repeat.
Sure. Does the CryoNet fold performance depend on the map resolution? And if you have any data, could you provide any statistics on that?
The CryoNet we develop is based on the public database, the data set, the data we collected from the public data, and for now the performance is for those density map with at a resolution higher than 5 angstrom, about 80% of the residue amino acid can be correctly modeled. But for those 3D density map at a resolution worse than or lower than 5 angstrom, about 60% of amino acid can be correctly modeled.
Okay, great. Thank you. And a follow-up to that, Pan, you had a question in the chat from Alex Noble. Will CryoNet be published and made available to the community at some point?
Oh, okay. The CryoNet have not published now, but we cooperate with Cliff Zhang, who is a scientific advisor of Shemu and also a professor of Tsinghua University. He published an online version with the website of cryonet.ai. Everybody can access this website to test the CryoNet.
Okay, great. Thank you. Next question is a little bit broader in terms of artificial intelligence in general. For you, Raymond, does Thermo Fisher have any plans to incorporate AI into any of its software?
That's a good question, and certainly if you look at the Cryo-EM workflow, there is a lot of decisions you need to make when setting up your experiment, your data acquisition experiment, as well as steering your experiment along the way. That requires a lot of expertise from the user. So we are actively implementing now ways of automation, and then sometimes it's non-AI based, but increasingly it's AI based and machine learning that takes away that, let's say, supports the user to make the right decisions or automatically makes the decisions for him. Yeah, I would certainly recommend people who want to dive into the details there to watch the June 29th product launch of Smart EPU. A little bit of advertisement, but there we will show our first real product that will help a lot of people, I think, making the workflow easier and hopefully also faster. I think it really also goes in line with the answer about the modeling and the data processing. We want to achieve, of course, the same thing on the data acquisition side. Main thing is just AI and machine learning in general. AlphaFold itself is a bit less relevant there. It's really the how do you mimic an expert at the tool without having him or her to be at the tool.
Oh, great. Yeah, and thanks for the ad. I didn't know about the launch in 3 weeks, so yeah, I'll stay tuned. Follow up on that for you, Raymond. What kind of computational power would you suggest for folks that are doing AI model building in their density maps? Like, what kind of system would you recommend for those that are getting set up?
I don't mean what kind of system.
Oh no, sorry. I was asking Raymond as a follow-up. But Pan, if you have a suggestion, that'd be great. If you have any recommendations.
Okay. The 3D density map, the input for our CryoNet model is the 3D density map and the query sequence. The model can use the sequence to search the MSA and the PDB templates, and this model is like the AlphaFold tool because we borrow the idea, the deep learning framework from AlphaFold tool to build the atomic model. Okay.
Great, thank you. Pan, maybe a follow-up for you. Unless Raymond, did you have any thoughts on suggestions?
Maybe one addition to that. I'm not so much into what kind of hardware is required, and I think the CryoNet is a great development. If you look at what a golden standard is now, I think you would be looking at Phoenix, and if people are interested, and most of them probably have watched Tom Terwilliger's webinar on how AlphaFold is implemented into the computational or the modeling pipeline. And I think that's a great way to look at how it can speed up And the next step is, I think, CryoNet, but that is already a great way to improve the modeling. I have no idea really what computational power is required for that.
Yeah, I think that's a personal decision, almost like people prefer Macintosh versus PC or Linux, etc. And so this follow-up question came from Kako Stapleton. For your AI-based solution for data processing, have you tried it with a benchmark protein? For example, what happens when you apply this approach? Do you get for beta-galactosidase, for example, do you see the same results as with the standard approach?
Okay. I think the best standard protein is the spike SARS-CoV-2 spike protein. Because this protein, our company has solved many spike proteins. Our program has tested on the spike protein, and most over than 95% of the amino acid can be correctly modeled.
Great, thank you. Thanks, Pan. Xiaodan, I wanted to bring it back over to you here. We've got a question about some other applications. How do you see AI being used perhaps in other areas besides single particles, such as tomography, micro-ED? Any thoughts from you?
Okay.
Yeah, maybe I can answer for my Pan, then Pan will have a supplement. So I think in my part, the most benefit I think is for model building. That is for sure. And the second thing I think is for protein expression. Yeah, sometimes because now use AlphaFold2, we can have a prediction of your protein, and you will know what the roughly structure looks like. And if it's a flexible region, for example, the GPCRs, we can based on the flexible region and do the protein design. And for Cryo-ET and micro-ED, actually, I don't have much experience for apply AI on both Cryo-ET or micro-ED, but I think the AI could help in data acquisition, probably. Yeah, the similar as SPA. Yeah, maybe Pan will have more comments. Yeah.
Pan, do you have any suggestions?
Okay. Xiaodan's answer is complete. I have nothing to add.
Okay, great, thanks. Let me bounce it over to one of our Thermo Fisher colleagues. I know Eddie works a lot with sample prep and some of our other methods. Eddie, any thoughts on you with AI regarding tomography, micro-ED, et cetera, going in the future?
I think, just to jump back to something that that Xiaodan then mentioned, in terms of other areas that AI might be useful in the structural biology workflow, the Cryo-EM workflow. I think one area is, and it was mentioned, protein production. And I think there's a wealth of data that exists, from the 15 years of the Protein Structure Initiative and all the work done at the SGC to just name some of it, and all of the internal work done at pharma companies, of looking at construct design to protein expression in various model organisms to protein purification. And I think that data is ripe for the mining to see if we can do some predictive modeling of if you input a sequence, can we get out on the other end what the best expression, what the best construct and expression conditions are. And I know there's a number of papers that have come out recently and a number of initiatives sort of undergoing a few that we're involved in looking at this. But that's just a wealth of data that exists from years that can be leveraged to sort of help the structural biology workflow. And then to get to the specific question that was asked, for tomography, for example, I know there's a lot of work being done looking at how we can use computer vision and AI to help sort of segmentation, let's say. For example, if you have your initial tomograms, and how do you do those first steps of image processing, and segmentation, I think is the, choose my words carefully, maybe low-hanging fruit of the workflow, but you could certainly expand on that. And I know, within here Thermo, there's groups working on it, as well as a number of papers coming out almost weekly in the tomography space of people doing something similar.
Yeah, tomography is going to blow the roof off. And talking about computational power, we're going to need some pretty heavy data servers to manage that. Thanks. Raymond, I wanted to bounce something over to you. A question came in about drug discovery, and maybe you could comment on how combining Cryo-EM and AI could help accelerate the drug discovery process, perhaps.
Yeah, that's the question of AI and drug discovery is obviously way bigger than only Cryo-EM, right? So with Cryo-EM and experimental structure determination, we need to fit in the drug discovery cycles. And if you look at small molecule drug discovery, where do we fit in? If you look at virtual screening, from billions of compounds down to a few, where you can use Cryo-EM to do experimental structure determination on these compounds and then go into an optimization cycle. And certainly there's a lot that we can add. And I think on the data acquisition side, we're at the speed that we can fit into those cycles. But of course, there is other parameters that have a lot of focus in drug discovery as well that are non-structural related, like the toxicity and other compounds or other properties. Sorry, I lost the question a bit. Can you please repeat, Brad?
Oh no, it was just combining Cryo-EM with artificial intelligence, and I think that the solution is it closes the feedback loop and it accelerates it, right? I mean,
yeah, so that's really for small molecule drug discovery, and that is very clear, right? I think for biologic, the story is a little bit different, but I think the fit in there and a close integration, and I think that's very important, the close integration of the experimental workflow with the in silico workflow, and where a lot of the AI lives, is, I think, essential. And yeah, if people are interested, we have a number of white papers on that we've put online, and more are coming, that show how important that integration is.
Oh, great, thanks. It seems like there's a lot of interest in CryoNet. So Pan, I've got another question for you, or a couple of questions on CryoNet. So this one is, are you planning to integrate CryoNet into existing external software? And then what are the key refinements that CryoNet Refine provides?
Sorry, could you please repeat?
Sure, I'll ask you the first question. So are you planning to integrate CryoNet Fold into the existing available processing softwares, the publicly available software?
Integrate the CryoNet to other software.
For example,
okay.
Make it more available or license it, etc.
Okay. CryoNet is CryoNet is used to build the atomic model from 3D density map. I think it can be individual software. Also, we are integrating the CryoNet Fold to the CryoNet platform. We are building the CryoNet platform. Platform means that we have many software in this platform, and CryoNet Fold is just a single software in this platform. Great.
I have a sample prep question. Maybe Eddie, you can help out with this one. This is from MD Karim, asking about what do I have to keep in mind making cryo-EM sample, for example, with PROTACs. So we know PROTACs are a hot area of structure now. Anything you could add to sample prep, for example?
Yeah, that's a very loaded question. So there has been, recently a few papers showing structural characterization both by X-ray and Cryo-EM of PROTACs, and I think, I don't have any sage advice I can sort of say right now that that answers that question, but really it comes down to sample preparation and make sure that all of the components of the system that you're trying to solve, that you have those, that you screen all of the various E3 ligases and everything, for example, to make sure that you have the right binding partners to have the PROTAC complex that you're looking for. And I think, sample stability and, these things don't like ice per se, so, sample stability during the vitrification process is a challenge. And so yeah, this is not a very good answer, but there's a lot of hurdles when you start thinking about PROTACs and sample preparation of PROTACs, and really it's making sure that you have the sample that a stable sample in the right E3 and the right components, and then really trying to optimize vitrification conditions as much as possible, and that usually involves the addition or the incorporation of additives and things like that to just make sure that sample that complex stays a complex through the vitrification process. So maybe others on the panel, maybe from Shuimu that have some experience in PROTACs can maybe comment as well.
Xiaodan or Pan, have you guys worked on solving some PROTAC structures?
We have some experience. Yeah, we have several experience about solving PROTAC structure, but it's for sure. The PROTAC is very flexible. Yeah. So I guess our client need to go back to a protein production, back and forth, and try to add some cross-linking. Yeah, cross-linker into the complex, try to make it stable. Yeah. So I think the stability is the most important thing for the PROTAC complex. Yeah.
Okay, great. Well, thank you. And everyone handled that minefield pretty well. I think I have another easier one for you, Eddie, and everyone can chime in here. And we already talked, this is from Nina Gonella, but we already talked about the lower molecular weight components, but can Cryo-EM handle membrane proteins? Eddie, I know you know something about that. Maybe you could start it off.
Wow, you go from a very challenging question to one right in my wheelhouse. I love it, Brad.
The best one, yeah.
Yes, absolutely. Cryo-EM is uniquely suited for membrane protein structural biology, and I could probably talk for hours on this, but there's what we've seen in the last few years, especially, is that Cryo-EM is the number of structures determined, unique membrane protein structures determined by Cryo-EM is far out eclipsed the number of unique membrane protein structures determined by X-ray crystallography. And there's just certain tools regarding sample preparation, for example, SMALPs and lipid nanodiscs and certain detergents that are amenable only really for Cryo-EM and not X-ray crystallography that sort of facilitate the sample preparation process. And so yeah, the short answer to that question is yes, absolutely. Cryo-EM is uniquely suited to solve the structures of challenging membrane proteins.
Yeah, and I know that the crystallographers, they're very jealous of that. Pan and Xiaodan, any comments, additional comments on membrane proteins?
Yeah, for membrane protein, I think Cryo-EM technology is the best way to do membrane protein, because the membrane protein is always resistant to X-ray crystallography. Yeah, I think, and we have lots of experience of membrane proteins, including GP-SAS, membrane transporters, and ion channels. Yeah, we actually, the first time we actually use different kind of combination of grids. As I mentioned before, we have the graphene oxide, reduced graphene oxide, and also the holy carbon or ultra gold. Yeah, so we always try different combination of grids, try to get the best quality of the sample. Yeah, so I think the quality of sample for membrane proteins is most important. And then secondly, we need to get a high concentration of a membrane protein. If you get a high concentration of your protein, means that you can have a very thinner ice. Yeah, I think the thinner ice and high concentration is a good combination to get good particles of your membrane protein. Yeah, so that is the second thing. Yeah, I think that is what I want to say.
Oh, great. And Pan, any final comments on membrane proteins, or do you agree with Xiaodan?
I have heard some people say that use the AlphaFold tool to predict the membrane protein. Some people said that the performance is good, but some people said that it's bad. So I have no idea.
Okay. Well, stay tuned. I think that's a round for AlphaFold 3, maybe. We've reached the end of our questions. Luckily, we've got through all of them. Everyone has done a great job on answering them, so thank you. And I tried to share the questions equally with all of you. But before we go, I'd like to thank the audience for joining us today and for all their interesting questions that we did not have time for. There may be one or two that I didn't see, but we will address via your contact info that you registered with. The webcast will be available to view on demand on our Ask the Experts webpage. And you can also go there to register for upcoming sessions. We encourage you to share this with your colleagues who may have missed today's event. And we're always looking to improve. So please submit any feedback after the close of this with the online survey. But I'd like to again thank you very much to our panelists, Xiaodan, Pan, Raymond, and Eddie once again, and especially everyone out there in the audience for joining us today. So until next time, thank you and goodbye.